Assalamualaikum and good afternoon to everyone. Thank you so much for joining our fifth series of Immb Research Talk for this year. So today we have Associate Professor Technologist Dr. Cheng Siang from UNIMAS. Dr. Tan will be giving a presentation on the introduction to biorisk management. Before we proceed, I would like to welcome our IMMB Director, AP Dr. Wang Siok Moy, to give a brief introduction on Prof. Tan. The floor is yours, Prof.
Thank you. Is there an echo behind? I think I can hear some echo,
Yes.
but I'm not sure where is the source of the echo. Perhaps.
It's not mine.
Okay.
But it's not much. It's not too much.
Oh, not too much. All right. So, okay. It's my pleasure to introduce Dr. Cheng Siang, Associate Professor of Virology at the Faculty of Medicine and Health Sciences, University Malaysia, Sarawak, UNIMAS. Dr. Tan currently leads both the Department of Paraclinical Sciences and the Therapeutic Research Center at UNIMAS. His research focuses on vaccine-preventable diseases, zoonotic pathogens, and the molecular epidemiology of emerging infections within a One Health framework. Dr. Tan holds a PhD in virology from Newcastle University, United Kingdom, and has authored over 30 peer-reviewed publications spanning virology and biosafety. He has secured numerous national and international research grants and is a recipient of both the Southeast Asia One Health University Network Fellowship and the National Science Fellowship. As a recognized leader in biosafety and biosecurity, Dr. Tan was among the first graduates of Malaysia's Advanced Biosafety Officer Training Program in 2013 and has since served actively as a national trainer. He holds 4 professional certifications from the International Federation of Biosafety Associations, including credentials in biorisk management, biological waste management, and biosafety cabinet safety. He also chairs the Institutional Biosafety and Biosecurity Committee at UNIMAS and has contributed to the development of Malaysia's national biosafety guidelines. Dr. Tan, the floor is yours.
All right, thanks, Prof. Wang. I've known Prof. Wang since my university days. So today I'm going to talk about the introduction to biorisk management. I'm not sure how many of you actually were trained in biorisk management. So today's talk is really, really basic. And I hope towards the end, you will be able to grasp hold of the concept behind biorisk management. Okay, next slide. So, okay, we stop here. Biorisk management, it has 2 components. One is biosafety, and another one is biosecurity. Next slide. And the definition of biosafety is a laboratory, it describes the containment principles, technologies, and practices that are implemented to prevent the unintentional exposure to biological agents and toxins and their accidental release. So you will see that the prevention is not only towards biological agent alone or the bacteria, the viruses, but it also mentioned about toxins. Most of the time, we overlook the danger of toxins. Next slide. So I just close down everything that is probably not that important. Biosafety basically is about unintentional release or accidental exposure. All right. So biosafety is unintentional. When you get into an accident, it is highly unintentional. All right. Next. So this is biosecurity. Laboratory biosecurity describes the protection, control, and accountability for biological agents and toxins. You see that we always mention about biological agent and also toxins. okay, within laboratories in order to prevent their loss, theft, misuse, diversion of, unauthorized access, or intentional unauthorized release. Continue. So go back. So the keyword here is intentional unauthorized release. So previously, when we look at biosafety, we are talking about unintentional. Biosecurity is about controlling the intentional but unauthorized release. Okay. So in the text, you also see that it's about theft, misuse, etc. Next slide. And in order for us to manage biorisk, there are two widely acceptable models that you can follow. One on the left is the AMP model, which stands for uh, risk assessment, mitigation, and performance. And on the right is the PDCA model, plan, do, check, and act. So these two, basically, they are the same. It's just a different author, they categorize things differently. So for the left side, the risk assessment, you have on the right, you have the plan. So you plan your experiments. Then when you have on the left, you have the risk mitigation. You do it, okay? You, you, you execute what you have planned. And towards the end, on the left, you have the performance, but on the right, you have the check and act. So you need to check what you have done and whether it works for you. Most of the time, we will use AMP model because it's more simple to use. Next. And the first step of the AMP, so now I'm going into the AMP model. So the first step is always about the identification of hazard. And the definition is any object which can cause harm. And the risk, once you can identify the hazard, then you can calculate the risk. So risk is actually a mathematical function of likelihood multiplied by consequence. Next. So I've been using this slide for many, many years now. I'll ask my audience, how many of you will go and participate in such an event? Some people said yes, some people said no, but how many? And obviously, for the person pictured here, he accepted the risk. If you analyze properly, uh, this guy, he's able to climb high, doing mountain climbing, rock climbing. He accepted the risk. He was very much aware of the hazard. That is, the gravity, the fall. And if it happens, the consequence will be disastrous. So how did he manage to avoid or minimize all these risks? You see that he has harness on, the ropes, everything that can break a fall in case an accident occurred. That will minimize the risk. And of course, you need to have some trainings, and you must have physical strength, etc., so that you are able to hold your weight. Next slide. So instead of just going into bacteria, viruses, let's look at this scenario. Diving, taking picture of a shark. How many of you would do this? So personally, I would not do it because I do not understand fish. I do not understand sharks. But some people would do it. And usually in a open open workshop, I will ask my audience, how many of you are willing or will take the risk to swim with a shark? So most of the time, probably one or two will raise their hand. But let's go to the next slide. The hazard actually is about the genus of the and the species of the fish that you are swimming with, the size of it, and the availability of food at that point of time, and whether it's a mating season. So swimming with fish, it doesn't mean that you must swim with shark, right? But swimming with shark, it depends whether you are swimming with a great white shark or other species. Next slide. And whether you can accept the risk of swimming with shark, it depends on you. Whether you're experienced in doing it or you have received professional training, you have the health to do so, whether you are complacent, and the general perception of the risk of swimming with shark. Okay, next slide. So we know the hazard here is a shark. And on the left side, we have likelihood. So it's categorized into almost certain, likely, possible, unlikely, and rare. And on the top, we have the consequences. When something happens, would the consequence be insignificant, minor, moderate, major, or severe? So this is the risk matrix that help us to make a proper risk assessment. And mind you, you see that at the center here, we have the low risk, the moderate risk in yellow, and then gradually proceed to extreme. So let's say a person is swimming with a great white shark, about that size that we saved just now. So the risk is getting bitten by the shark. And although the cases is possible or rare, the consequence can be major or severe. So you see that the risk itself is high to extreme because you can die from it. Next slide. So I will ask the audience again. In the second picture, would you now reconsider to swim with shark in the catch? In the normal situation, I would see that some of my audience will now raise their hand because I feel protected within the catch. This is a kind of mitigation procedure. So you keep the shark out and then you keep your person in. But the risk remains there. You cannot eliminate the risk because at times you can, if you happen to feed the shark and your hand is outside the cage, then there is always a risk of being bitten. Okay. Next. But we take into consideration the swimming with shark in a cage. And usually those who have raised their hand, if I ask them, if you have watched one of these movies the night before, would you still proceed to dive with the shark in the cage? Some will say no, because despite the risk remains the same, but now their perception of the risk has changed because of external influence. So what I'm trying to bring here is sometimes you see that certain thing is extremely dangerous. It is not. It is because of your background, your exposure, and your training. Okay. Next. So the third picture and the fourth picture here, it shows you do not have to swim with a shark, with or without a catch, because there's always a better way to do things. The safer way to learn about shark, that is watch a documentary or don't go into the water at all. All right. Next. So to do a biorisk assessment, we do the the 5 Ws, the what, why, when, who, and where. What is biorisk management? It's basically a systematic process of evaluating the potential risks that may be involved in an activity or undertaking. So remember that it is a systematic process. It is not about your perception only. And why are you doing it? Because it forms an integral part of a good occupational safety and health and biorisk management plan, and determines if existing control measures are adequate or if more should be done. And when do you need to do it? This one, sometimes as scientists, we sort of disagree. Because when do you need to do biorisk management? It's really before you initiate your work, before you even start. So you need to plan ahead to see whether you are able to mitigate the risk to an acceptable level. And who should be performing the risk assessment? It should be done by those who are most familiar with the activity or the project or equipment that is being accessed, or who are the people creating the risk. And where should it be done? It should not be done in my lab. If it's your lab, your work, then it should be done within your facility, among the experts in your facility. In case where there are no content expert in your facility, then it is wise to seek external help. Okay. Next, other than the 5W, now we go into how do you need to perform biorisk assessment. I introduced this HACCP, or the hazard analysis critical control points. If you have heard of this, it'll be good. If you have not heard of this, you should Google it up and read about it. So the way I perform risk assessment, I always use the HACCP approach. Okay, you will have to break down everything into its the protocol, and then you have to look at the risk that each step will likely to generate. So, for example, the serological screening of blood for parasite X from monkey. This is a case study here. So how do I do it? Most of the time, if you are not well versed in biorisk management, then you'll be looking at parasite X, perhaps the monkey. Other than that, you may not have much worry about handling or sampling parasite X from monkey. Next slide. So using the concept of HACCP, then I can break it down to the different steps. So the first step is you have to go to the jungle, entering into the jungle to look for the monkeys. Secondly, is to capture the monkey. The risk of entering the jungle, it depends on who is going in. If you are bringing a zoologist, an experienced zoologist, they know the jungle inside out. So it is fine. But if you are entering a jungle that you have never been before, then the risk becomes higher because of other hazards. And to capture the monkey, it really depends on what kind of method that you use. And the third one is securing the monkey for phlebotomy or blood draw. So who are performing all this? Are they trained? Are you sedating the monkey? If yes, then the risk will be lower. And then the phlebotomy procedure, storage of blood, and how do you centrifuge the blood, the process of pipetting, and ELISA plate washing, and disposal of waste. So you have to break it down into different SOP and look at the risk itself. Next slide. And like I mentioned just now, when we perform risk assessment, it could be the very first time that we handle pathogen, or it might not be even be a pathogen, just a microbes, a bacteria, and or a non-infectious virus, a virus that can infect animals but not human. So, uh, infectious agent itself, when you perform risk assessment on an infectious agent, you need to consider the agent itself as an individual. All right, like for E. coli, not all E. coli are pathogenic. The one that can cause diarrhea or to a certain extent hemorrhagic or septic shock, there are pathogenic strains. So if you are obtaining samples from the hospital, these are of higher risk compared to the one that you use for cloning experiments, right? And then you need to understand the reservoir of the pathogen, how it's being shed into the environment, the mode of transmission, the portal entry, how you can be infected, and the susceptible host. So some pathogens, they are not, uh, they cannot infect human. So in this case, despite the high transmission rate, it will not affect human. One example is if you are working on African swine fever. It, it is highly infectious among the pigs, but unfortunately, fortunately, it doesn't infect human. Even if you get exposed, it won't establish an infection. So this kind of knowledge help you to make a proper risk assessment. Next. This was taken from WHO for risk group classification of the various pathogens. So previously, pathogens were classified into risk group 1, 2, 3, and 4 based on few criteria. Like for risk group number 1, I will say this is a food grade kind of bacteria or pathogens. It has very low risk to human as an individual, and it won't transmit to the community. Even if it transmit to the community, nothing happen. It won't cause any disease. Risk group 2, these are normal human pathogens. And if it if you get exposed to this pathogen in the lab, you might get sick, but not really sick. Okay, it is not fatal. So the risk to individual is moderate, colored in yellow orange here. But the transmission to community can be low. Okay, the risk to community is low. And risk group 3 normally is associated with higher risk, and also um, and risk group 3, it can be pathogens like um, Nipah virus, etc. When there's an exposure in a laboratory, the risk or the consequence to the individual is high, but subsequent transmission to the community can be low. All right. And bear in mind that if you read about biosafety level or the BSL 1 to 4, that this is a classification of different laboratory facility. And risk grouping has nothing to do with biosafety level. So it doesn't mean that risk group 3 agent must always be done in the BSL3 facility. Okay. So on this slide, you know that when you do risk assessment, you need to consider individual and as a community. Next slide. Since the emergence or since the term One Health has been coined in, so we do understand that the risk of transmission is not only to human, it can be to animals like livestock, to the community, and also in the environment. So when you perform risk assessment, you need to consider everything here. If a pathogen that can establish a reservoir in the animal, like SARS-CoV-2, then the disposal of waste or anything related to the pathogen, it needs to be controlled. So the mitigation procedure must be there. So the next time when you consider to do risk assessment, then you must consider the risk or the consequence when it's not being decontaminated properly and exposed to the animals, even plants, and the community. Next slide. I'll give you another scenario, a bit like the shark. So I hope you can understand uh, the same hazard, it can pose a different risk to the handler. Next. Scenario 1. You have encountered with an aggressive and hungry tiger while trekking in a jungle. So from your side, you just assess the risk. What is the likelihood of you being attacked by this hazard? And then, if so, what is the consequence? The keyword here is the aggressiveness and the hunger of the hazard. So it is likely or almost certain that the tiger will attack you, and the consequence will be severe. Next slide. And you encountered with this well-fed and caged tiger at the National Zoo. What is the likelihood of you being attacked? Now you see that the risk and the situation has changed. The tiger remains the same. Even if you can break out from the cage, the likelihood of it attacking you is low. So likelihood can be low, or it can still be possible. If it is possible, then the consequence will still be severe. All right. If you just visit the zoo, then it is unlikely or rare that this tiger will attack you. The next slide. So this is the scenario 3. You are bottle feeding a cute tiger cub. So probably this is the tiger, the cub that has yet to achieve maturity in scenario 1. But in this case, the likelihood of you being bitten is rare. And if it's so, the consequence will be insignificant. So it is actually very low risk to handle. All right. Next slide. Let's go more into the laboratory side, the mycobacterium scenarios. Next slide. So you are a medical lab tech culturing a sputum sample on the Lowenstein-Jensen medium slant. So the hazard here is mycobacterium from the sputum, primary specimen. So the likelihood of exposure, I think, is pretty low. But it also depends on where you perform the streaking. And if the consequence, what is the consequence if it happens, it is still be quite major. So the potential risk is medium to high. Okay. But if this mycobacterium is sensitive to all antibiotics, then the risk can be lower. Next. You're a postdoctoral researcher culturing Mycobacterium tuberculosis in 50 ml of liquid culture for plasmid maxiprep. So in this scenario, the hazard remains the same. but the person operating it is now different. It's a postdoc, but I did not mention uh, what kind of postdoc, whether he has years of experience in mycobacterium handling, or he came in from another discipline. And the 50 ml liquid culture is actually a lot. And to do a plasmid maxi prep, you need centrifugation. Centrifugation can introduce aerosol, and the inhalation of the aerosol potentially can transmit a mycobacterium to himself or to people working in the lab. So the likelihood of transmission, it is possible or is likely, and when it happens, it can be either high to severe consequence. Next slide. So you are a scientist at a pharmaceutical company, and you are preparing a culture of attenuated Mycobacterium bovis for vaccine production. Mycobacterium bovis can to a certain extent elicit immune response in human, and that is why it's used as a vaccine strain. And in the event where Mycobacterium bovis is being released into the laboratory environment, uh, the consequence is actually insignificant or very minor. So the risk can be either low or medium. So now, uh, for these two scenarios, I want you to appreciate that not you have to look at the pathogen and the person who operates it as a whole. You are not supposed to look at the pathogen mycobacterium. You can't do it because it is dangerous. That is very subjective of who is doing it, how you perform the experiment, what experiment you are doing. So we have seen biorisk assessment, and also, sorry, we are we have done the biorisk assessment side. So let's look at how we can control or mitigate the risk. Let's go to the next slide. I'll introduce you the hierarchy of control for the mitigation procedure. So I'm going to give you an inverted pyramid, and the first one is elimination. So elimination means you don't do it. Upon risk assessment, if you think that the risk is too high, and all your colleagues and your institutional biosafety community confirm that the risk is too high for you to handle, then you can say, no, I'm not going to do this research anymore. If your laboratory is not a BSL-3, then culturing Mycobacterium tuberculosis can be of high risk. So you can say, no, I don't want to do it. Then if you don't do it, there is no risk. Okay, that is the only way that you can eliminate the risk. Next slide. Let's say you are researching into the pathway or the binding of a virus, a dangerous virus, to the host cell epitope. So instead of using the actual virulent pathogen, are you able to substitute it with the vaccine strain or the attenuated strain? So if yes, then this is what we call the substitution. We change it with something that is similar but less virulent. In this situation, you are able to reduce the risk significantly by using something safer. So in SARS-CoV-2 research, sometimes we use what we call the pseudovirus. We use a less virulent virus to express certain protein or certain structures of SARS-CoV-2 to study about the binding of the pathogen to certain cells. So that one would significantly reduce the risk. And the third one is engineering. Engineering is something that people love. Like one thing that you must have seen in your lab will be a biosafety cabinet. So biosafety cabinet allows you to potentially generate aerosol, but not getting yourself exposed because it handles the aerosols within the biosafety cabinet. And nowadays, we no longer aspirate a pipette. We use pipetters. We use partial pipette. And all these things, they are engineering innovation. And for biosafety level 3 laboratories and other uh, laboratory facilities, these are classified as engineering controls. You can have a directional airflow installed. You can have HEPA filter. You can have different sensors being installed in certain equipment. You can have ultraviolet light installed. These are forms of engineering control. Next. So as we go down the pyramid, you see that the effectiveness of controlling the risk is getting less effective. Administration is about training. Like today, I am giving you awareness on the introduction of biorisk management. So if you are not aware, then you are not able to make proper assessment and you are not able to do things safely. So administration is about control, controlling your, sorry, administration is something like the SOP. So you establish SOP for your lab tech to follow, your student to follow, because you believe that this is the best way to do it safely. So the SOP are there for this purpose. And having SOP, eventually you will have a lot of paperwork. You have different files for different protocols. And over the years, new students will come in, new staff will come in, they may not know the existence of such protocols. So training and retraining is required. So this is what we classify as administrative control. Okay, and there are times when you need to submit hard copy of your risk assessment, a formal risk assessment to the Institutional Biosafety Committee for approval. This is a form of administrative control. Are you able to start your research without approval? If yes, then you have eliminated administrative control. So administrative control is very important. So there are risks that you think you can accept, but it is actually very dangerous to yourself, very dangerous to people around you, or even your institution. So why you need institutional approval? There are accidents in laboratories here and there. Sometimes it reaches a headline, sometimes it's not. But when there's an investigation of laboratory acquired infection, especially on the severe side, then the first question is, did this researcher obtain institutional approval to carry out this experiment? So if you do it quietly, then you're on your own. Okay. And we go up one step, that is the engineering. Is the engineering control sufficient for certain pathogens that you are working on? Okay. The last one. The last one is personal protective equipment. This is something that we always love. We like to use masks, we like to use glove. In actual fact, this is the only barrier before your skin, only barrier before you inhale in air. So the effectiveness is the worst, the lowest here to control uh, the hazard. Okay, and the proper use itself is administrative. You need to be trained how to use glove, how to remove it. You need to know which respirator to use, when to use it, how to remove it. So all this is a mixture of administrative and PPE or personal protective equipment. Controls in biosecurity. I'll just summarize it in one patch. So physical security is important. So just now we talk about identifying hazard. We talk about the risks pertaining to certain procedures, certain experiments, and we talk about how we can mitigate all these risks. And the second one is how do you secure it? I would like to bring you to the point number 3 here, valuable biological materials. This is the legal term used to refer to all valuable biological materials. You can be working on pathogens, and these pathogens, it has risk of being stolen to be used as a agent of bioterrorism. So that is one point. But valuable biological materials is a bit wider. The scope includes vaccines, vaccine strains. You might be developing a vaccine against Ebola. So this vaccine strain becomes a valuable biological material that, if stolen, people can actually benefit from it. Other parties can benefit from your invention. So how do you keep all these things, the pathogens, toxins, even protein that you have synthesized, secure? So number one here is a physical security. So you see that in most laboratories nowadays, you have the key and lock, and you have control access using thumbprint, using smart card. You can use the retina scan and so on. This one is really to control access to your facility. So do you have such a physical control in place? Then the second one is actually the personnel. So how much do you vet through your personnel? When a staff comes into you applying for a job, do you actually screen their background? Do they, when they claim they're on their CV, they are experts in microbiology? Uh, is it true? Did you actually contact the previous employer or the university where he or she graduated from, that all these informations are indeed true? Or is this person affiliated with uh, terrorists in other countries? So personnel becomes an issue because there will be instances where uh, high-risk countries, they send their spies or their scientists to certain countries to learn and sometimes to steal. So do we know our people in the lab? So most of the time, theft, terrorism, uh, it can be caused by or start from internal job. So the vetting of personnel that have access to valuable biological material is relatively important. And when it comes to valuable biological material, you need to keep a record of where this VBM go to. What we have is a material control and accountability. Who owns this material and for what purpose? So there must be some legal uh, tracking system available. For instance, I asked a vial of virus from Dr. Wang. And since I know her for a long time, she just gave it to me. And in actual fact, that is part of a commercialization project. But I also use the same strain to commercialize my own stuff. So there is a conflict now. So when you give samples to people, you must have this material control agreement and or material transfer agreement MTA, specifying what I can formally do or use this sample for. So this one will keep your valuable biological material secure. And information security is a last control in biosecurity. So what kind of information are you storing in your computer? And how secure is your internet or intranet access? Can everyone actually access the computer in the lab? Do they need a password? So this is called information security. In laboratories where the risk is high or there's a high value in their valuable biological material, especially in commercial labs, then people cannot bring in or upload data into their thumb drive because you can steal the biological information from there, right? So this is the end of the AMP model. So the performance. Performance is all about measuring the effectiveness of your plan, your assessment, and the mitigation procedure. So is there an opportunity or a platform for you to perform periodic reporting? And who do you report to? Especially when there's an accident, especially when there's a potential exposure, or when there are near misses. For example, I drop a vial of pathogenic bacteria on the floor, and then it smear all over the floor, and some droplets can be seen on the wall. That's an accident, but I might not have been exposed. So can I actually report this to someone in your institution? If yes, then we will need to review what happened. So probably I was carrying the vial or on my hand or in a plastic box without a proper secure design. So the annual review or the protocol review will need to be reassessed or reviewed annually and then be improved. That is why if you remember back the AMP model, it is a triangle. After you do the assessment, you do the mitigation, and then you do the performance. And after you do the performance, you will do the assessment once again, every year as a minimum. Next, what we have learned. So I like to use the iceberg as an illustration. So what we learn here is very minimal. And at times, where after mitigating a risk, we thought we have done everything. But in the next slide, go to the next slide. We really do not know a lot. So we do need to do annual review. The AMP model must be active. It must be done periodically, at least once a year. And sometimes when you perform some experiments and you are okay, you still alive today, it is not because you did not get exposed. Accidents can have happened. You might be asymptomatic. So what we learned from the COVID pandemic is a lot of people with COVID, they are actually asymptomatic. So does it mean that it is right to be exposed in the laboratory, but you're still okay? The risk is low, but something wrong had happened. All right. So assessing the risk in a periodic way can actually help you to understand what is underneath uh, the sea level. Next, I would like to end my talk and open the floor for any question.
Thank you so much, Prof. Tan. For the online audiences, if you have any questions, you may type them in the chat box or unmute yourself.
Thank you, Prof. Tan. Okay, thank you, Prof. Tan, for introducing us the basic of biorisk management. Okay, I have a question. If I were to bring, for example, a vial of viruses uh, to UNIMAS, for example, across the sea, so do I need to apply any permission?
There must at
But
least be a
yeah,
MTA. The
MTA,
airline must be
sorry.
the
Uh,
airline must be aware that you're carrying something. If it's a pathogen, then the Ministry of Health, there's a pejabat. Let me see. There's a. I have the letter. You wait for a while. There's a pejabat kesihatan KIA that needs to clear your stuff. So you shouldn't be shipping things here and there, even within Malaysia.
within Malaysia. All right.
Yeah,
So this is under the Ministry of Health, or we need to apply the permit from Ministry of Health or Ministry of Transport. I can't hear you clearly just now. Yeah,
Ministry
the
of Health.
Ministry of
And
Health. All
yeah,
right.
but then the packaging will be done triple packaging according to the IATA standard.
All
If
right.
if you are to ship something dangerous, I would prefer you to use a reputable courier system that can handle or that will train to transport dangerous goods.
Okay. Can you give some example of reputable courier system? I think we have used a DHL previously. Yeah.
Uh, there is there are two companies. One is definitely World Courier. They will do all the permits for you. And another one, I believe, is GSSM. It's a relatively new company, I think. I can give you the address if you want later.
Okay. Noted. Thank you. Thank you, Prof. Tan. Okay.
You can ask questions pertaining to your research if you want.
Do we have other questions?
Or anything that you are not clear about my presentation.
I have one more question, Prof. Tan. If, for example, if there are a mini outbreak near my campus, if by collecting the specimens, can I conduct the isolation in a BSC2 biosafety cabinet?
Yes, you can. You are allowed. Let's use the guideline is like this. You are able to perform isolation or other mitigation procedure in BSL2. You can do the diagnosis, all this, but once you find out that, wow, there's a Nipah outbreak in my campus, that one you need to stop work and move everything to inform MOH, the Ministry of Health, and eventually all your samples will be transported to, I think, NIH or the previous IMR. If you do not know, then it's an environmental sample. Then you can do it in BSL2 facility.
All right. Thank you for your explanation, Prof. Tan. Yeah.
Yes, Mohaimin, do you have question for Prof. Tan?
Hello, Prof. Okay.
Hi.
Hi. First of all, thank you so much for your interesting sharing. I just have a question. Actually, I'm a postgraduate student from
Okay.
Faculty of Medicine. So my question is, because I'm working with COVID-19, okay, given the evolving landscape of this infectious disease that I mentioned, like the COVID-19, and we know that as time, the implementation of vaccination, some people got vaccinated and so on. In your opinion, how often should we actually conduct the biorisk assessment to manage and maybe they mitigate the biological risk? Uh, because in this example, it's a COVID-19 lah, but then maybe in the future, we don't know some other viruses as well. Yeah.
How frequent, let me recap your question.
Okay, okay.
How frequent do we need to perform risk assessment or the AMP using the AMP model
Yeah, yeah.
on your work
Yeah,
in
maybe in your
COVID-19?
in your opinion lah, or we should or we can actually just stick. For example, when I were to start my project, I need to apply for the biorisk assessment and so on.
Okay.
But maybe in future, for example, if I'm working, because for now I'm working with the virus-like particles, which is a safer alternative as the actual virus lah. Then I
Mhm.
applied for the biorisk management. For
Okay.
example, in future, uh, uh, I believe I still need to apply for it. But then, in your opinion, how frequent should this actually the assessment being done, Prof?
The assessment, okay.
Yeah, yeah.
In biorisk management,
Mm-mm.
the key, the best answer is it depends.
Mhm, mhm,
If
mhm.
the risk is very low, then you can actually extend the duration. But
I
if
see.
the
I see.
risk is, if we know it's really nothing,
Mhm, mhm,
nowadays the risk of handling SARS-CoV-2 is extremely low.
mhm, mhm, mhm,
Okay, most of the time, or nearly all people, they get cough or asymptomatic, so the
mhm,
risk is actually low. But for the sake of administrative process, risk assessment or the review of your protocol, everything should be done annually.
mhm,
Because
mhm,
biorisk management is actually a culture. If you
mhm,
say this person, researcher one, is doing it every 2, 3 years or per project, then how come I need to do it annually?
mhm,
So
mhm,
I'm sure along your experiments, you learn something that you have done is suboptimal.
mhm.
So it should be documented and then your protocol should be updated.
Okay, okay, all right, all right. Okay, thank you, Prof.
Anymore?
Thank you for the questions. I think there is no other questions. So on behalf of IMMB, I would like to thank AP Dr. Tan Cheng Siang for the informative sharing and knowledge, as well as the online audiences. Thank you so much for joining. Next week, we will have a Fulbright Scholar student, Joseph Kasali. So please also join our talk next week on Wednesday from 12 to 1. There will be a physical as well as online talk next week. For this talk, I will upload the recording to YouTube and I will share the link later. So thank you so much to Prof. Tan and also everyone for joining our talk today. Please don't forget to fill in the attendance form. I've shared the link on the chat box before leaving. Thank you so much.